Hello and welcome to my channel Happy Learning to You, your ultimate guide to easy learning. So let's continue with the topic of genetics under this chapter of principles of inheritance and variation, and the topic we are going to discuss today is monohybrid cross. So let's get started. Well, this is what the diagram which I am showing here is something which I have explained in my previous video. You can have the link above, but still let me explain it back here. Now, what Mendels experiment involved was crossing between a tall parent and a dwarf parent, which ultimately led to the F1 generation, wherein it was observed that both the offsprings were tall. The characteristic of only one parent, that is the tall parent, was expressed at the F1 generation, and the dwarf character of the other parent was not expressed. Later, what he did was he carried out self-pollination of these tall F1 generation plants, and what he observed was in the F2 generation, 3/4 of the plants were tall and 1/4 dwarf. So the characteristic of this parent, which was not expressed at F1 generation, was now expressed at the F2 stage. Now, if we use alphabetical symbols for each gene, then we shall use capital letter for the trait which is expressed at the F1 stage, and small alphabet is used for the other one which is not expressed at the F1 stage. So as per this, a capital T is used for the tall trait and a small t is used for the dwarf trait. Now, as you know, genes that code for a pair of contrasting traits are known as alleles. Basically, alleles are slightly different forms of the same gene. So if you consider the definition, then this capital T and the small t are alleles of each other. Now, what Mendel suggested was not to use a capital T for tall and a small d for dwarf, considering the first alphabet of each of these, because then it would be difficult to remember whether this T and D are alleles of the same gene or not. So for better understanding of the alleles, he suggested to use a capital and small letter of the same alphabet. Okay, next. Now, what he proposed is that in a true breeding tall or a dwarf pea plant, the allelic pair of genes for height are identical. Okay, so as per him, a true breeding tall pea plant, the allelic pair of genes was capital TT, identical. And for a true breeding dwarf pea plant, the allelic pair of genes were small tt. So, this descriptive word, that is tall and dwarf, these descriptive terms are known as phenotype, whereas the alphabetical terms is known as the genotype. So please do remember in all the upcoming videos what you mean by phenotype and genotype. So phenotype is the descriptive term and genotype is the alphabetical term. Now, since the capital TT and small tt, here these two allelic pairs are identical. Both the pairs here are capital and both here are small. So these are known as homozygous. Whereas when one is capital and one is small, it will be known as heterozygous. Now, Mendel observed that the phenotype of the F1 heterozygote TT, because heterozygote is the allelic pairs are non-identical. So the phenotype, that means the descriptive term, the description whether tall or dwarf of this F1 heterozygote is exactly like the TT parent, that is the homozygous parent. So the TT, as per this phenotypes, that is the TT heterozygote was tall, which is equivalent to TT. So as per description, if you go to consider, then this heterozygote and this homozygote both were tall. So the phenotype was same, but genotypically they were different. proposed that in a pair of dissimilar factors, one dominates the other. This is known as the dominant factor and the other is a recessive factor. In this case, the T for tallness becomes the dominant factor and small t for dwarfness becomes the recessive factor. Because tallness is the dominant factor because this TT has phenotype, that is the description is tall. So it gets expressed as a tall plant and not as a dwarf plant. So therefore it is a dominant trait and the dwarfness is a recessive trait. Now he observed similar behavior for all other trait pairs that he studied. So as you know, Mendel had carried out the study on 7 traits. Okay. Now since the TT plant, a capital T and a small t plant is heterozygous for genes controlling one character, in this case height, it is monohybrid. And the cross between the capital TT and small tt is known as a monohybrid cross. So this is all I have under the topic monohybrid cross. In the next video, I will explain about the use of Punnett square in order to understand the monohybrid cross. So Punnett square is basically a tool which is used to understand this monohybrid cross, how this crossing occurs and ultimately how you get the capital TT or the small tt. Okay, so do keep watching this space for more videos on genetics. Thanks for watching and do subscribe to my channel for more educational videos. Thank you.